just go ahead to move on. I want to first review the RNA sequencing part. So today is the third lecture. And the first slide here it shows you the RNA sequencing, the general pipeline. And this is the messenger RNA. And then the first step will do the RNA or cDNA fragmentation. Okay, it depends on where your reverse transcription happens. If you do the reverse transcription first, you will do the cDNA fragmentation. If you do the RNA fragment, after the fragmentation, and you probably. So the point here is these sequencers so far only working on the DNA. At a certain point, you have to change that from RNA to DNA. Although there are some technologies that are trying to work on the direct RNA sequencing, but not there yet. And next step after the fragmentation, you want to do the library preparation, and you can see the linkers there, and then you do the sequencing, and you got those ACGTs. And from there, you want to go for the alignment part, bioinformatics analysis. This will align the reads back into the genome or transcriptome. And you can see there's some reads there called axonic reads, and they aligned on the axon regions. There are junction reads, and there are polyadenylation reads. And then you can further do whatever other analysis that you need to do. And traditionally, when we talk about the transcriptome analysis, the first thing that jumps into our mind is the gene expression. And we know that which gene express and by how much. That's important. But RNA sequencing allows us to go to the transcriptome in a higher dimension. And the second one is the splicing patterns. So you can see there's one gene has 3 exons, and in one condition, it can both all the 3 in the final product. In the other condition, only the middle one is skipped. So even with the same gene, same expression level, you may end up with a different biological product. And the third dimension is the variation on the transcript. Even this gene has the same expression level, same splicing isoform, but because of the single nucleotide variation, that can cause further consequence, including the protein structure alterations. Okay, we'll get into this a little bit this lecture. And what we can do, get out of RNA sequencing experiment, this is a summary slide. You can see the RNA sequencing can give us the gene expression and further figure out the differential expression, and can do the alternative splicing, and can identify the transcriptome variation, including the non-synonymous variation, synonymous variation, variations on the 3' UTR, or further allele-specific expression. And for the synonymous variation, it could disrupt the RNA binding, protein binding, therefore affect alternative splicing. If the variation happens on the 3' UTR, which, by the way, you can identify those variations in the RNA sequencing data, and if they are microRNA binding sites, that will further affect the differential expression. And another advantage of RNA sequencing can give us the non-coding RNA profiling, which sometimes hard to do on the arrays because we don't know too much about where those non-coding RNAs are. I'm not going to go through this array principle anymore, but the limitations of a microarray platform, the first one is the high background level because of the cross-hybridization. So we cannot really measure the low expression genes very well. And also because of the saturation, we cannot measure the high expression gene very well either. So the dynamic range of microarray technology is much lower, and this is a limited dynamic range part. And another limitation is arrays rely on existing knowledge about the genome sequence. We have to know there's a gene there to put it on the array. Sometimes we don't know exactly what we know and what we don't. So RNA sequencing doesn't have to have those type of prior knowledge in terms of human genome. And this is one from a paper that published in 2009 summarized the differences between arrays and RNA sequencing. And the point I put a circle here so you can see the background noise for the RNA sequencing is lower, and so that you can go through this, and the dynamic range are higher, and things like that. So you feel free to go through those. For the experimental considerations, so when we prepare for the samples, this is not too much to do with the analysis part. So we talk about the amount of total RNA that is required. Initially, it's a very large amount, which goes to 100 microgram. And but people has the protocol, you can even work on the single cell level. So the dynamic range of this part is pretty high as well. And based on GNAT, that our university, the system so far is comfortable working with maybe 500 nanogram, of total RNA. That is pretty good. And we mentioned the importance of ribosome removal because 95% of the total RNA are ribosome genes, and we don't need those. We don't need to know their expression levels. And so there's a significant need to remove those. And otherwise, all our arrays, 95% of our arrays goes through ribosome, which we don't want to see. The reverse transcription, there's two ways to do that. Either use oligo-DT or random primer. And they both have their advantage and disadvantage. If you do the oligo-DT and you get rid of ribosome RNA perfectly, because ribosome doesn't have the poly-A, and but you are restricted kind of on the genes that are polyadenylated. There's a lot of non-coding RNAs, they are not polyadenylated, they will be missed. Another problem of using oligo-DT is the 3' end bias, because those RNAs will be degraded, and every single molecule measured has to have the 3' end to be able to reverse transcribe. So you will see much higher signal towards the end of the gene if you use oligo-DT only. And you can use random primer, but you have to go through those ribosome removal part. And we also need to consider whether the assay itself maintains the strand of the RNA. Sometimes when you study non-coding RNAs, it's very important to have those maintained because we don't know the exact annotation of those non-coding RNAs. And other thing we need to consider is the parent end, whether you want to use that or not. If you believe gene fusion, for example, is something that you want to discover, you really want to look at in your sample, probably parent end is a must. But sometimes for other type of applications, you may consider the balance between whether we use the parent end or we want to save money. So by saving money, that you can run more samples using the same cost, and which also increase your statistical power. So those are the things that when you design the experiment, you have to explore a little bit. We went through several different published studies. The first one we went through is the mapping and the quantifying of a mammalian genome. This is kind of the first RNA sequencing paper that was ever there by the Barbara Wu's group. And then we also mentioned how to use a paper to study alternative splicing using RNA sequencing from Chris Birch's lab. The gene fusion is another study we mentioned, and the mRNA sequencing whole transcriptome analysis of single cell, and you can see that people can work on that level. And genome annotation for the non-mortal organism, because for some of the species, we don't have the reference genome, so we don't know too much about the reference genome, so you have to use RNA sequencing to annotate those. And various in the complex disease, and that is another important application. So these are the things that we went through last time, and also the small RNA profiling, which we will get into hopefully a little bit today regarding their data analysis part. After introducing those applications, and we started to look for the analysis part. The first analysis tool that we need to go through is the alignment. And we know that RNA sequencing alignment is different from DNA sequencing because all those short aligners, they do not allow big gaps, okay? And we know some of the RNA rates because we are sequencing the transcriptome, and the junction rates, if you align it back to the human genome, and you will miss those. So some of the people ask me after the class, what's the percentage of rates that map to junction? Can we just ignore those? But think of the length of the rays, let's say 50 base pair or 75 base pair. Do we have an idea how long the exons are? Do we have an idea? Nobody? General statistics? Give a guess. Let's say median value. What's the median value for the nucleotide length of exon? 1,000 base pair? 100 base pair? It's 127 base pair. And if you based on that kind of statistic, of course some are shorter, some are much longer, and 1/3 of the 50 base pair rates will be on the junctions. And as we are talking about today, that those junction rates are really, really important for you to study alternative splicing. And So those are something that we have to have special consideration. So in terms of splicing alignment, there's two general approaches we talk about. The first one is called the exon-first approach. So basically what you do is you align those reads back to the genome, so you can see those exon reads has been aligned first. Forget about those hybrid ones, which is the junction reads. And then you further using different type of strategy to map the remaining reads back on the junctions where your exon has been expressed. And the second type of, which is much less popular, is the seed extension approach. So first, you separate those reads into smaller windows, and then for each individual window, you map them and join them back together. Okay? And the most popular approach to do the RNA sequencing alignment is called top hat, and that is a typical exon-first approach. Okay? And after the alignment, the first question, we still want to see the first dimension of this transcriptome, which is the gene expression, and we talked about it last time, which is sometimes to calculate the expression levels of the gene is a tricky issue as well. The major thing is because of the alternative splicing. And you can see that this is one gene, and there's two different isoforms. So one isoform, you can see this exon is missing, and so the dark blue one. And another isoform, and this one is there, and these two are missing. So there's alternative splicing. There's which reads you really need to use when you're trying to calculate the gene expression levels. The general approach are exon union or exon interception methods. For the exon union methods, we are using all the exons. The reads fall into all the exons. And the good thing of this is, of course, you use more information. All the reads that fall into this region, this gene region, you're using it. And the problem of that is that it underestimates the expression of alternative spliced genes. So this is one typical figure. You can see that this is the true RPKM, and this is the estimated RPKM by the dark blue is using the exon union approach. You can see that it's consistently under-evaluating on their expression levels. And the second approach is to use the exon intersection approach, which only focus on the constitutive exons and forget about alternative spliced exons, only using the reads that to those exon regions. And the good thing of this is it gives an unbiased evaluation of the gene expression, but the problem is you are using much less information. For those reads, you don't get those. Okay? But nevertheless, for the RNA sequencing gene expression levels, keep in mind exon union approach and exon interception method. And after we get through the gene expression levels, we want to calculate the differential expression. So you get two conditions, and which gene differential expressed between two conditions. And there are many methodologies that have been developed for array-based studies using read coverage to quantify gene expression levels. Sometimes we can use those techniques, like t-test or things like that, as long as your sample size is bigger enough. Or we briefly mentioned the Fisher's exact test statistics, or t-test based on the generalized linear model, identify the gene expression estimation. If you don't know what that is, go back to your notes to look at that. And the model, the biological variability in the absence of large number of biological replicates. So if we don't have 30, 40 samples in each group, and at that time we have to go back to the original distributions of the data. And we know that gene expression levels for the microarray, roughly we think of after doing log transformation, it follows normal distribution. But for the RNA sequencing data, it more follows Poisson or negative binomial distribution. So how to use those distribution equations to help us to increase our statistical power. And the last thing we want to keep in mind, the factors affecting differential expression and the quantification strategy really also affect the differential expression call. And we had example in the lecture. And some of the genes, if we use the exon union approach, we don't see any differential expression. If we use the exon interception approach, we do see it. And the sequencing length and library preparation, flow cell effects, and GC contents are, all those things are part of the game. So you need to consider those as well. Okay? Any questions so far? Perfect? There will be at least 2 quiz questions out of those review. Just some of the hint. Actually, we got a busy schedule today. We will talk about alternative splicing, gene fusion, allele-specific expression, RNA editing, and small RNA sequencing. Okay, and most of them are conceptual. I need you to understand it, not really go back detail into the equations and which step is doing this differential, integral, and things like that. Don't worry about that. But I want you to have the general understanding of what is the biological information being used, what's the assumption of it. Okay, let's first talk about alternative splicing. And the number of genes in the multicellular organism, you probably see these slides before. And we know that in human gene, there's about 20,000 genes, human genome. And the last time we talked about in C. elegans, which is the worm, we have about 18,000 genes. Okay? So there's only 1,500 genes that we are more than worms. Okay? So one of the jokes I always give is we have to find out some molecular mechanism to explain that we are better than the worms. Okay? So in terms of the number of genes here, and they don't really distinguish too much. And alternative splicing here is one mechanism that sits on top of genetic code and contributes to the biological complexity. And alternative splicing is a process where one gene produces more than one type of mRNA. So you can see 3 exons, and after different conditions, and all the 3 exons in final product, and only the middle one has been spliced out. And this can be tissue-specific and condition-specific. And those could have very serious phenotypical effects. And alternative splicing is an important level of gene regulation and major source of proteome diversity. And we know this deregulation, if they are not controlled well, and we will be sick. There's a disease, or especially cancer-related. And we also know about 90% of human genes express multiple isoforms. So this is not one gene or 2 gene issue. And it's estimation that over 60% of disease-causing mutations disrupt splicing. So when we go through this figure, you can see that there's many different types of alternative splicing. Not always like I plotted 3 exons and the middle one is missing. All right. There's many different types. So the first type is called the cassette exon, which is the one that I plotted. The middle one can be missed or can be there. And the second type here is the alternative 5' splicing sites. So you can see that when we talk about the directionality of the splicing, we always sit on the introns. Okay? That's why this site is called 3' end, and this is called the 5' end, not on the exons. All right? So this is the alternative 5' splicing. So you can see in one condition, the splicing happens in this way, in another in this way. And alternatively, you can see the alternative 3' end variation. And there are also other types. This is a mutually exclusive exon, and this is intron retention. So the middle part can be completely spliced out, can be leave in the final product. And there's also other 2 types of events that are also you can consider as alternative splicing events, alternative events, but they are not happening on the splicing stage, which is alternative promoter. So you can see that the start of the gene can be different, or alternative polyadenylation. That means their end is different. But the end results of all these events are you have one gene have multiple isoforms, but some of them, the top ones, are regulated in the RNA level, and for this alternative promoter, it's regulated in the DNA level. So that's the major difference. And actually, there's for different tissue types, there's also very different occurrence of all these events. So the estimation is in human brain, has brain and the liver, or the 2 organs, have the large amount of splicing isoforms. Okay, and bone is pretty bad. not too many isoforms. But for the liver and the brain, it's very active in isoforms. And the brain, the most isoform happens in the cassette exon phase, and for the liver is in the alternative 5 and 3 prime. So those are the things that it's good to know. And the reason when we study alternative splicing, of course, is because of the splicing patterns. They are complicated. They can be they can have phenotypic effects. Another thing is there are something that is regulating this splicing. This including the RNA binding proteins, RNA secondary structures, and other factors so that cause this. And the one thing that you commonly see from literature is there are cis-acting RNA elements that is regulating the alternative splicing, and the ones that are on the axons and in the introns. And some of them is promoting the splicing, others are inhibiting the splicing. That's why you see this ISE, ESE, or ESS, ASS. Those stands for exonic or intronic splicing enhancer or silencers. The point here is there are RNA binding proteins binds in somewhere in the precursor mRNA and then regulate the splicing. Before we have the RNA sequencing technology, and there are some technologies that we can use to genome-wide identify the splicing patterns. The first one is the axon array, especially Affymetrix axon array. So we know that traditionally when we talk about microarray technologies, we use a probe to target each individual gene. So this probe targets gene number 1, this probe targets gene number 2. Okay? We are using gene as a unit. Okay? But for the axon array, instead of using the entire gene as a unit, they are using each individual axon as a unit. So that by the end of the day, you can see this is the Affymetrix array design. For each axon, they design up to 4 probes to target each axon. Therefore, you not only get the entire gene expression levels, but also each individual axon expression levels. And this is one of the examples. You can see the middle axon. This is one gene, multiple axons. In one condition, it's low across the board. And in another condition, it's pretty high, but it's low for this particular axon. That is really suggesting that there's alternative splicing occurred. Okay? And after the axon array technology, and the axon array technology is perfect when you think about that. But when you really do the experiment using it, and we are pretty miserable. And I was very much miserable. And there are some better technologies and to increase the sensitivity of splicing detection. One of the things is also array-based technology. It's called splicing junction arrays. So instead of targeting different axons, they are targeting different splicing events. Okay? For example, this one is called the axon intron retention, right? So for one isoform, this is the axon, intron will be spliced out, and another one, this is not spliced. So what you do is you design probes to target all different locations here. So most importantly, you got one probe to target on these junctions. And if you see the difference that between the probes signal supporting the splicing or supporting the inclusion, and then you will know that which part the splicing happens. The point here is splicing junction arrays, you are designing the probes to target on the splicing junctions that help you to identify alternative splicing. And it turns out that this technology has much, much higher sensitivity. However, in order to design these arrays, we have to know the species in terms of potential alternative splicing pattern really, really well. Okay? You are not guessing there's an axon here. You know there's an axon here. You know there's a splicing could occur there. That's the only way that you can design the probes to target on that. Specific sensitivity, I mean, the data will look very, very good for the ones that you're targeting, but you are potentially missing a lot of other things. Okay? Does that make sense? Okay. All right. Now let's go through the RNA sequencing technology. And using next generation starter splicing, again, this is a simple scenario. So you can see there's many different rays here. And all different rays, after you do the proper alignment, there are some rays, like the black rays here, okay? Those are called constitutive axon rays, constitutive rays. That means those rays really locate on the constitutive part, and it doesn't change across both two different isoforms. And there are some rays, which is a red ray. They support the inclusion of this event. Think a moment. So this axon is an in or out. If it's in, and those red rays will be identified. And if it's out, the green rays will be identified. So you can see that one end on this axon, the other end on this axon, and the middle one has been spliced out. Okay? So there's three types of rays: axon inclusion, axon exclusion, and constitutive axons. And these are the most important information that we need to use to identify the alternative splicing. And we have seen this example before. So this is in a real life example. There's an axon. There's so many axons here, and then there's introns. And this particular ray is supporting that this axon will be included, and this ray supporting this axon will be included, and this ray supporting that this axon will be excluded because these two axons, they join together. And this will tell us that there are multiple isoforms in this particular gene in the sample. Okay? So now let's go to the analysis part. The computational strategies for that is, the first one is called isoform-based approach. So what that really means is, this is the RNA sequencing data, expression levels, you see the rays and things like that. And this is two different isoforms. The problem here is the isoform information is invisible for you at the moment. You only see the distribution of those sequencing rays. You don't really know the isoform at this stage, right? And then what's your goal to do this RNA sequencing is trying to see, to reconstruct those isoforms. That's one of the goals. And so it's called isoform-based approach. For this particular example, you can think of probably this isoform has about 20% in the final product, and 80% is this isoform, because the middle one, the expression level, is much, much lower. And if you're smart, you can figure this out, right? However, once we got multiple isoforms on one gene, it's pretty difficult. You have to have strategies. Sometimes it's very rare, it's very common that one gene has 10 different isoforms, and at least potential isoforms, and how you can reconstruct that requires some computation. And in terms of isoform-based approach, there's two smaller categories. The first one is called the genome-guided reconstruction, and we will spend most of the time to talk about two tools. One is the coupling, and the other is the scripture. And the second one is genome-independent reconstruction. So basically what you do here is you first do the assembly and then do the isoform reconstruction. So we'll get to there. But both these two general categories are isoform-based approach. And there's another way to look at this problem is from the event. So as I mentioned, if you have multiple isoforms here and you have problem to reconstruct those isoforms, but if you're sitting on this alternative splicing event, you are facing only two choices. It's included or excluded, okay? Everything else doesn't matter. You're sitting here, look at this particular event. So you can use the individual alternative splicing event as your unit. And then for this particular example, you probably think, oh, 20% of the transcripts contains this axon, 80% doesn't, right? So if we have only two isoforms, no big deal. Okay, they are the same. But if we have 10 isoforms, okay, and the isoform-based approach is very difficult. But for the splicing event approach, I'm still having that or not having that. And my final evaluation will be what's the percentage of final transcript have this. Okay? So there's two general approaches in terms of alternative splicing. So let's go through. First one is the genome-guided reconstruction, and there's two very, very important tools. Okay, if you study RNA sequencing data, use this technology. This is something that everyone needs to know, especially the first one, the cufflinks. So let's go through that. Just the basic principle, no mathematical equations for this. The first step is you need to do the alignment. And you have to choose a aligner that is capable of producing spliced alignments. And top hat will be a good choice, right? We spent a lot of time last lecture talk about the strategies to do the RNA sequencing alignment. We have to pick the right one, okay? So you can do the top hat and you do the alignment. You can see that there are some of the reads. And I mean, this is the pair end. So basically, you sequence both two pairs, two ends of a transcript. And for one connection, if you're seeking from the back, you probably won't see it, but look at your notes. And if it's a solid line connected, that means that is one fragment, and you have two ends sequenced. And for this one, that you can see that this is a solid part, actually. This is the ones that was not sequenced. But this is the dash line, and this part is the junction part. So one read is across the junction. And you will see this is spliced fragments alignment. So this is after alignment, you will see this. The next step you want to do is to identify a minimum set of incompatible fragments that must have originated from different spliced mRNA isoforms. And for this data, this schematic example is these three reads. There is no way they are from the same mRNA isoform. Okay? Let's think of why. For these two, okay, you can see that for this one, and the middle axon is included for sure. And this one, the middle axon has been excluded. So these two reads is not going to be from the same isoform. That makes sense? All right? So because of the difference here, right? And this read is not going to be compatible with either of this because clearly that this in this isoform, this part expressed, and for this part expressed, but in these isoforms, they were both spliced out. Okay? And this is a minimum set of incompatible fragments. They are not compatible. You can find another set of incompatible ones. This is the minimum set. So, for example, this one, and this one is not going to be compatible to each other as well. Fine, if that's your choice. Yes?
I have a question. So what's the solid and the dotted line mean?
Solid is, so this is a pair end. Okay, look at me on there. This is the paired end. So you got RNA fragments, you're sequencing both two ends. All right, the middle one is not sequenced, but they are kind of connected. The dashed line here is for this one end, all right, it's across the junction. All right, so after they are connected in the RNA level, but once you map it back to the DNA level, they are separated because there is a junction occurred there. That makes sense? Okay? All right. So the second step is very critical. Identify a minimum set of incompatible fragments from different isoforms. Yes?
Usually when they try to, so the LB is your comfort maximum set of compatible fragments.
A maximum set of incompatible fragments.
We can have all that.
And you're lowering the specificity then. So we'll get into that. So the question is, if we use this part, and we probably will identify something real, but we are also running into the risk to missing something. Okay, we will miss some because they are not really. We say that these two are compatible. It may also from two different isoforms. Who knows, right? And so we are in the risk of missing something, and we'll talk about next approach is fixing that part, but in the other part we don't like as well. Yeah.
So in the minimum size, I have also size that will have a good number.
Right. anyone is fine. So that's what I said. If you are choosing this race, and this race, that is okay. Okay, that is okay. So we'll look at the next step. So this is the minimum incompatible race. And next stage is trying to make a graph, connecting the dots. So here is, you can see that all those rays, okay, and I put the middle one, and I define this as a node. So each fragment corresponding to a node here. And the edge will be the connecting two different fragments. I only connect them if they are compatible to each other and their alignments overlap. So let's see what that really means. For example, this is the fragment, this is the fragment, and I'm connecting those together because they are compatible with each other, and they can occur on the same isoform, and also they are overlapping. The same thing happened here. So this one and this one, they are compatible with each other, and they have some overlapping. But I'm not going to connect the yellow one with the blue one because they are not connecting to each other, because they are not compatible with each other. And I'm not going to connect this one with this one, although they can be on the same isoform, but they are not overlapping. Okay? So the first stage is to identify a minimum set of incompatible fragments, and the next stage is to make this graphic and to connect the individual dots, individual fragments. That make sense? Need some more time? Yeah.
Oh, I guess for example, in machine learning or other usually it's done like that. This minimum set serves as an initial starting point.
Mm-hmm.
So different initial starting points may lead to different.
Mm-hmm.
Do they actually converge to one final result, or?
That's a good question. I don't know. I don't have the answer for that. I guess for this particular example, you may. So the question is, for machine learning, if you start from different initial parameters, and you start one initial parameters, you're converging to one set of results. You start from another set of parameters, you may converge to that result, maybe converge to something else. Whether the final product is sensitive to your initial choice of these alignments, I don't know. I have no answer for that. Okay. And you may try more complicated approach. I mean, data sets and to give a test. I have no idea about that. All right. So it's okay. I say no, right? Because I really don't know. Next step is to construct the path. So you can see that there are different. So we already connected the dots, and then you just conceptually construct it and to merge those. Those are one path. There's another path and third path. Okay, and then using this minimum path cover and to get to the transcripts. So you can see there's three potential transcript isoforms. The first one representing the yellow one, which is the one that this part is spliced out. And for the blue one, so you can see there's a small leftover in here, and then there's a potential exon in this region. But for the purple one, it's most likely to be not spliced at all. All right? Is that all right? Okay. So the overlapping graph can be minimally covered by three paths, and which is these three colors. And the next step you're doing is for these three different isoforms, you want to estimate their transcript abundance for each different isoforms. So you can see those colors is labeled there, and together with which potential isoforms they can belong to. So clearly for the yellow part, and this one is the yellow one, right? And if the blue one, this is the blue one, and the purple one. So all the rays that support this red one, a purple one, it's labeled as this same color. And there's one thing that you cannot see very clear is this one, this rate. Is that right? Let me see. Yes, this rate. So this is, keep in mind, this is a paired end. This is a paired end, okay? And for this particular rate, it can be from either this isoform or this isoform. So you can see their color is labeled a little differently. Okay? And for all these rates, they can be from all the 3 different isoforms, so they are labeled as black. Okay? So what you want to do at this stage is you map those individual rates to see which individual isoforms it's compatible with. And then together with the fragment length, when you do the RNA sequencing experiment, because you know that both 2 ends, how long the middle RNA fragments is, there will be a range of that. And then running that through these equations, which I can tell you some of the parameters being used here. Let's forget about the equations. Rather focus on what's the information in the equation. The first one is this row. This row is actually the one you want to identify. It is abundance of different transcripts. Those are a parameter. And this is a Bayesian type of approach, so you're trying to calculate the likelihood of the row given all the read information. And another information being used here is the length of the transcripts, and there will be a distribution of the length of RNA fragments. Okay? So this information will be used together, and eventually you just solve this complex equation and lead to this maximum likelihood abundance estimation, which you can see that the purple one expressed this much, and together with the blue one and the yellow one. All confused? Go back to look at the notes after the class. So the summary slides for the cufflinks is it requires the splice alignment, such as a top hat, and it's capable of identifying and quantifying isoforms from RNA sequencing data. Okay, not only identify the isoforms, but quantify it. And it can be used both on the single end and paired end. And there's a concept that here is for the pair end, it's called the FPKM, not RPKM. Okay, RPKM stands for reads per kilobase exon model per million mappable rates. But here, because of its pair end, and it's kind of FPKM, it's called fragments per K per M. And there are some following algorithms for the cufflinks. It's called the cuff-diff, and can help you to identify the differential expression of 2 different isoforms in 2 different conditions. Okay? Are we okay with the cufflinks? So conceptually, what I have said is you first do the alignment and then identify a minimum set of incompatible rates and then build the graphic. That's the trick. Okay? As Yang mentioned, we may miss something. There may be some isoforms. It's beyond these 3 isoforms. But these 3 isoforms are a minimum set. Okay? And it's all open source software, and I don't think we will go through another homework, but you can try if you like. All right. And the second tool that I'm going to give is called a Scripture, and this is it's kind of interesting. Is they published in the same issue of Nature Biotechnology with the cufflinks, just a back-to-back paper. And this is so cufflinks is it's developed in Caltech, and this Scripture is from MIT. And the first step is very similar. It's alignment, and this is the same as cufflinks. You also need to pick the alignment that's capable of producing spliced isoforms, okay, as top hat or things like that. So you can see that the figures is if there's, you will see some of the rates here, and there are some junction rates that connecting these 2 parts or things like that. And your second step will diverge a little bit. This is kind of very straightforward. Okay, this is nothing, no equation, just a strategy. At this stage, they only use the spliced rates. Okay, they only use the spliced rates. This is the part I hate. Okay, because we know it's hard to really do the splicing, the junction alignment is so hard. And sometimes we are missing things there, but at this stage, they only rely on those rays. Okay. So what happens is, and they will, only based on splice rates, they will make the directions determined using splice set sequence features, A, G, G, T. So you can see they will make this separation. There's one exon here, one exon here, and if there is a splice read supporting this, they will connect this to two. And if you can see, this is a conceptual exon here, and if there's one read that across this part, they will connect these two. So they will connect based on the splice reads. And it's a very strong requirement for the alignment quality, which is really, really difficult. And next step is they will construct a connectivity graph of individual base, still only based on the splice reads. So nothing special. They just make circles instead of this straight line. I don't know why they make it an extra step for that. But there is something special is you see there are directionalities here. So for this one, it's from another gene which is connecting, happens to be the same connecting together, and they are in a different color. So that's what they did. And next step is start to make sense is they will score each individual pass. So you can see those passes, and they will score each individual pass. What they do here is they will use both spliced and unspliced reads. And if they have enough enrichment here, and they will connect this. So they will, for each individual path, they will calculate the p-value based on the abundance of spliced and unspliced reads. And if the number is small, like 0.01, they will have cutoff, and that is more likely to be a real. For this one, it's 0.5. It will not be in the next stage connection. So you can see that those are the ones, and this is 0.8. It has been ruled out. So they'll just be considered as noise. And next step here is they will identify the number of graphs in this figure. And you can see there's one graph here, and there's one graph here, and there's one graph here. They identified 3 graphs with schematic figures. And this score significant path rather than exons increase sensitivity. And the last step is they will join these graphs. So you can see that this is a couple of separate graphs. This is one graph, this is another graph, another graph, another graph. But they want to join that using the pair and the raise information. So if they see there's a one read that cover this both two ends, they will join the graphic together. And what they do here is alternative spliced isoform identified by considering all the possible passes. And Yang, you will like this. So they are using all the possible paths in the transcript graph. So hopefully you are not missing anything. And eliminating isoforms that result in pair and reads map to a distance with low likelihood. So if there's an isoform making, if you look at the one pair and one fragment and two ends are too far away from each other, it's unlikely to be the case, and that will lower the probability that they're joining together. But anyways, they will evaluate those. And a summary for the scripture is it's also highly rely on reads across splicing junctions, and it's very strong requirement for the alignment. And longer reads will certainly help. So for this part, you need longer arrays. Otherwise, it's really hard to get through. So major goal here is for the transcriptome construction, not quantification. That is the difference. One of the major differences between scripture and the cufflinks. The cufflinks can do both the isoform reconstruction together with the quantification. For the scripture, you only do the reconstruction part. It doesn't do the quantification. And it's great for study regions without any annotation if you do the alignment correctly. And it requires pair end to discover isoforms. Okay, so everything I talk about cufflinks, scripture, I know that you have some idea, but you are not really having any idea how to do it at this stage. And I hope I encourage you to go back to look at that. And it's not that difficult. And if not enough information is provided in the slides, go back to the papers to look at those. But this is one slide I want you to keep. There's no quiz on this slide, by the way. But this is one slide I really want you to get the information on. What's the difference between the cufflinks and the scripture? Okay. They have a commonality. The first commonality is that they both are genome-guided reconstruction. So you have to do the alignment first and then reconstruct it. And both heavily relies on splice alignment. So you have to be very, very careful at the stage of doing alignment. You want to pick the one that is the best. Okay? It doesn't have to be the fastest. It has to be the best, because the end result is so much relying on this the alignment quality. The major difference, cufflinks is a minimum path cover. So maximizing what is maximizing is the specificity. Okay, meaning that if I see it, it's there. If I see it, it's correct. Scripture is considering all possible paths in the transcript graph. It's maximizing the sensitivity. Okay? If it's there, I have a higher probability to see it. But what I see is not necessarily correct. So you always want to find out the balance. It really depends on what kind of biological application you are doing and how much money you have for validation. If you want to only validate a few, you probably want to pick cufflinks, because every single thing you see is potentially real, but you're missing things. Here, you have a lower probability to miss things, but not everything you see are real. This is the information I want to deliver in this message. That's the comparison, in my view, of the cufflinks and the scripture. Okay? And are we okay? Let's take a 5 minutes break.